Good afternoon, or everybody, and for some of you, good morning. And so, welcome to the December webinar. We are extremely lucky that we've got Professor Bishr Omary from Rutgers University, who will present his work on therapeutic approaches for the treatment of epidermolysis bullosa simplex. Now, before I give you a brief introduction to Bishr, quick little bit of housekeeping rules as usual. Your attendees' cameras will be switched off and you'll be muted. The webinar will be recorded, so it means you can watch it at a later date on our website. Please submit your questions through the Q&A function. I will answer them at the end. The chat comments would also be enabled. And as always, please leave us feedback. We love that positive feedback. So if it's positive, even better. And if it's not, still leave your feedback for us, and then we'll find ways of improving it. And on that note, I'm going to pass it on to Bishr. But before I do, I'd just like to say Bishr is a professor of biomedical sciences at Rutgers University and senior vice chancellor of academic affairs and research for Rutgers Health. Completed a PhD training at the University of California, San Diego, and did his medical degree at the University of Miami. Spent 19 years at Stanford University and moved to Rutgers University in 2019. His lab studies the pathogenesis and therapeutic approaches to rare diseases that cause liver and skin damage, including epidermolysis bullosa simplex. And on that note, I will pass it on to Bishr.
Thank you so much, Sagar. Can you hear me okay?
I can hear you perfectly. You probably need to share your screen.
Let's see. Do you see one screen or do you see a split slide?
I see one screen, so it's absolutely perfect.
All right. Thank you so much, Sagar. And good morning, good afternoon, maybe even good evening. Thanks to all of you who joined this presentation. I think when Sagar was saying good morning, he was also referring to me as one of those folks. It's about 8 o'clock, a little bit after 8 o'clock here on Eastern United States time. So thank you so much, Sagar, for the invitation, opportunity to present some of our work related to epidermolysis bullosa simplex. So I'm talking about EB. I'm going to be talking about simplex, not going to be referring to dystrophic junctional or Kindler EB. And see if the slides are moving forward. So I'm going to try to touch on three kind of related topics. One is just to give a brief overview of intermediate filament proteins and their diseases, then review some of our drug screening to identify small molecules that normalize EBS mimetic keratin mutations. And what I mean by mimetic here, actually, I'm a gastroenterologist by training, so we worked on intermediate filaments specifically, or primarily, I would say, keratins and simple type epithelia, like in the digestive system. And it's really the work in the liver that took us to work on skin keratins from a therapeutic perspective, as I will cover. And then I will, last part of the presentation will be address preclinical development of a potential EBS therapeutic that we're very excited about. So one disclosure, and that's we do have a pending patent application that's related to the use of formulations and methods for treating EBS and related keratinopathies. So intermediate filament proteins make up a major component of the cytoskeleton. This is a platinum replica of a fibroblast. So if one can imagine taking a cell and kind of squeezing it, a fibroblast in this case, and stretching it out, what one sees ultrastructurally is a bunch of fibrillar elements that some of them, if you can see my pointer, are the inner aspect of the nucleus, and then these nice filaments that extend from the nuclear periphery to the cell surface. So that's one kind of working definition of the cytoskeleton. It's made up primarily of three major protein families: microfilaments, the major proteins being actins; microtubules, of course, tubulins; and intermediate filaments. There's a whole bunch of them, as I will show you in a minute. So I'll just touch on unique aspects of intermediate filaments, which include the keratins, as you will see. Their structure is quite diverse as compared to microfilaments, microtubules. Their distribution is limited in that they're only found in higher eukaryotes. They're quite insoluble. It depends on the intermediate filament protein. I'll touch on that a little bit. There are really no known stabilizers or destabilizers. For example, for microtubules, as you know, Taxol and Taxol derivatives are actually used to treat a number of different cancers. And speaking of diseases, their disease association is quite extensive as compared to the other two major cytoskeletal protein families. And in terms of function, they have several known, and the list is growing, and I will touch on this. So just by immunofluorescence, if one stains specifically, hopefully you can appreciate that these three cytoskeletal elements are quite distinct in their distribution, as shown in the triple staining here. Size-wise, ultrastructurally, the microfilaments, those filaments are approximately 5 to 7 nanometers. Intermediate filaments are 10 nanometers, and microtubules are 10. And their distribution of intermediate filaments, the nuclear lamina, nuclear intermediate filament proteins, sit just beneath the inner aspect of the nuclear membrane. The cytoplasmic intermediate filaments extend from the outer nuclear membrane to the plasma membrane, as you can see here with this double staining of lamin and a cytoplasmic intermediate filament protein. One important aspect of intermediate filaments from a biophysical perspective is that they're quite flexible, as you can see for the abbreviation IFs. Whereas, for example, microtubules are quite stiff. So, for example, they can easily break if you just try to bend them even as large as 1 millimeter in size, as compared, for example, to the much more flexible intermediate filaments in general, where they're quite flexible, as you can see in this cartoon here. So I mentioned intermediate filaments make up a large family of proteins. And there's 5 major types that exclude the orphan type. Two of the types are made up of keratins, and their distribution, just like the other intermediate filament families, is distinct. So all epithelial cells express at least one type 1 and one type 2 keratin, and they're obligate heteropolymers. So those nice filaments that I showed you one example of their staining, those filaments stoichiometrically, depending on the cell type, have half of type 1 keratins and half of type 2 keratins. If there's any leftover type 1, type 2 keratins, these are rapidly degraded in cells. So the ratio stoichiometrically is 1 to 1. Others are cell type specific, and they're used a lot by pathologists. Vimentin is a marker for mesenchymal cells. Glial-fibular acidic protein is a marker for astrocytes. Desmin muscle neurofilaments for neurons, and so on. The lamins, as I mentioned, make up the nuclear intermediate filament proteins, and then the lens have 2 family members called CP49 and valencin. We had reviewed intermediate filament proteins about 20 years ago, and at that time, there were about 35 specific diseases that are either caused or predisposed to by variants or mutations in intermediate filament genes. Now the list is more than 30 different human diseases, and there's still some orphan in this case, I'm going to call it intermediate filament proteins that are not linked to a specific disease. And the explanation, one at least hypothesis, is that they're likely to predispose to disease rather than cause disease per se, but those are still under intensive study. So with the keratins, I mentioned there's type 1 and type 2, and another unique aspect, just like families of intermediate filament proteins, the keratin composition within different epithelial cells varies. So in the skin, which is a major topic, what we're going to be talking about, the suprabasal keratinocytes express primarily keratins 1 and 10. They also have some keratin 2, 11, and 9. Corneal epithelial cells have 3 and 12. As you can see, the different epithelial cell type. I think this presentation will be shared by Sagir. So basal keratinocytes express keratins 5 and 14. I will be talking about those a fair bit. We got into this business of intermediate filaments by stumbling across keratins 8 and 18. And this is something I've been working on for the last 30 plus years. Now, simple type epithelia, which are really single layered glandular epithelia, consist of these keratins. The major keratins in these types of cells is 8 and 18, but the only adult cell that expresses only 8 and 18 is actually hepatocytes. Other epithelial cells, like for example, biliary epithelial cells, whether it's in the pancreas or ductal cells, like in the pancreas, or biliary cells in the liver, gastric epithelia, which are simple type epithelia, intestinal epithelia. They also have some of these other keratins that are listed here. So keratins are abundant proteins in epithelial cells. So in simple type epithelia, like in the digestive system, for example, they make up anywhere between 1 to 5%. In cell lines, they tend to express a little bit more in terms of stoichiometrically keratins. So they're about 2 to 5%. If you look at the basal keratinocytes, the keratins make up about 30% of the total protein of these keratinocytes. Suprabasal keratinocytes express about 70% of their proteins are K1 and K10. And you can see them, for example, this is simply immunoprecipitation of keratins 8 and 18 from a colonic epithelial cell. And this is by Comasi stain. So we're talking about here, microgram quantities that could be immunoprecipitated, which make up about 5% of the total protein. The issue with keratinocyte keratins is that they're quite insoluble as compared to the simple type epithelial cells, which has made their study difficult, and it actually helped us a lot accidentally in terms of studying the regulation of these proteins, because they're much more soluble, relatively speaking, compared to the epidermal keratins. So they're used a lot in practical terms, medically as tissue markers, for example, to distinguish epithelial type tumors and undifferentiated cells versus non-epithelial, for example, mesenchymal, because that is important for clinicians in terms of deciding about what potential treatment to use when the cancer is undifferentiated. They're also used in a lot of clinical trials, particularly in the liver, as a marker of apoptosis, because essentially all intermediate filament proteins, except for the type 2 keratins, are substrates for caspases. So when a cell undergoes apoptosis, there's a specific cleavage site that cleaves right almost in the middle. And also there's usually 1 or 2 cut sites in the intermediate filament proteins, and there's specific antibodies. In this case, it's M30 that is used in a ton of clinical trials as a marker for apoptosis in epithelial tissues. They're also intermediate filaments. Another kind of general property, if you will, is they're found as inclusions. That's also used a lot by pathologists. So, for example, one marker of fatty liver disease, whether it's related to alcohol or metabolic syndrome, for example, is associated with formation of these keratin inclusions, which are basically amorphous deposits that are double stained here with keratin, the keratins and ubiquitin that show this double staining of yellow. They're seen by pathologists as also because they're quite large, so they can be seen by standard hematoxylin and eosin staining. And initially, they were described by Professor Omary, who's a surgeon, wrote this classic textbook published in 1914 that described, and this is actually a picture that was taken from this textbook, and you can see even more than 100 years ago, that picture is pretty much very similar to what people were seeing by immune staining 30 or 40 years later. I mentioned that this is also generally these types of inclusions are really markers of different diseases. So, for example, the so-called Rosenthal fibers are seen in Alexander disease, and also they're seen here ultrastructurally, for example. Muscle disorders, so-called desminopathies that are caused by mutations in desmin, are also associated with formation of desmin inclusions that may be related not only for desminopathies, but also for disorders that affect muscle biology that leads to disorganization of desmin proteins and post-translational modifications that lead to formation of these inclusions. Another important feature that really relates also to to EBS is that these keratins also function as stress and survival determinants in the acute setting and also in the complex setting. So it's all kinds of stress, mechanical stress, and all sorts of non-mechanical stresses. Heat, for example, in the mechanical and the skin of EBS, just simply rubbing the skin in the more severe cases can induce blister formation. Heat just being outdoors, infections, any kind of genetic stress with genetic modifiers, other toxins, and also intracellular stress, be it osmotic, for example, oxidative or metabolic stress. And all these kinds of stresses can lead to inclusion formation. For example, I mentioned the Mallory-Denk bodies in epithelial cells, inclusion-like formations in EBS, for example, because these keratins 5 and 14 aggregate, and also, or you can get disruption of the filaments, as I will be showing you. So just to briefly summarize, this is really years of work by a number of laboratories, including others. We've done quite a bit of work trying to understand the function of keratins in simple type epithelia, digestive organs. And there's a lot of evidence that basically supports the importance of these proteins in targeting. If you remember, simple glandular type epithelia, they're polarized. So they have an apical or luminal surface, and then they have the blood side or the basolateral side. So, and specific proteins are targeted either to the apical or the basolateral side. Another important property of these proteins is that they're highly phosphorylated in the context of stress by a number of different stress kinases. Because they're so abundant, as I mentioned, they really act as a phosphate sponge. And we showed this, for example, by mutating in transgenic animals phosphorylation sites of keratin 18 or keratin 8. And these animals are much more susceptible, for example, to injury, oxidative injury, toxins, things of that sort. And in the case of EBS, mechanical integrity is one important function of these epidermal keratins, but they also have non-mechanical properties that are kind of similar, shown in this cartoon. They are important, for example, in organelle topography and function, particularly mitochondrial function. Pierre Coulomb, who works a lot, actually is one of the discoverers of that mutations in keratin 14 leads to epidermolysis bullosa simplex, showed a few years ago that in keratinocytes that the keratins are important also in protein synthesis. So speaking of mutations in keratins, so Pierre Coulomb was the first author while he was working in Elaine Fuchs's lab. There were two papers that were published very close. One describing mutations in keratin 14 as the cause of EBS, and this is really the first disease association of this large family of intermediate filament proteins with human disease. I mentioned the 70 different diseases, but this is really the first one. Genetics approaches were used in Dr. Epstein's lab to show the association of K5 and K14 with EBS. And also Birgit Lane published a paper just a few months later in 1992 showing a mutation in keratin 5 as a cause of hereditary blistering skin disease, namely EBS. And thereafter, there was really just a whole unfolding, if you will, of association of diseases of intermediate filament proteins. The next pair, if you will, were keratins 1 and keratin 10 that were published both in Cell and in Science, and then the list just goes on and on. So I think for those of you who are on the clinical side and see patients, you're very familiar with this type of a picture that is seen in the most severe form of EBS. There's also other blisters, depending if, and here you can see more in the less severe form in the soles of the feet, and that's because of direct again stress, weight bearing. And this is just an example of mutations in keratin 1 and 10 1 and 10 that are associated with hyperkeratosis. So going back a few years ago, Namon Koo was a postdoc in the lab. She generated the first actually point mutant overexpression of an intermediate filament protein, and that was specifically keratin 18. And the reason for this experiment, because these are the proteins that we were studying, and we tried to understand what is their function, and we thought generating a mutant protein that is targeted to the normal expression cells, namely simple epithelial cells, which is a marker, which K18 is a marker of in mice, might unfold a function or a disease association. And in this case, the answer was yes. The function, for example, when one does liver perfusion, these cells are highly susceptible to death. So normally, if one does a liver perfusion, 5 to 10% of the cells are maybe die just during the liver perfusion, 95% are alive. But when you perfuse the livers of these mice, 95% of the cells actually are dead simply by the pressure of the liver perfusion. And went on to show that they're susceptible to all kinds of challenges, toxins, drug-induced liver injury, other kinds of liver stress. And the key finding with this mutation, which really is homologous to the most severe K14 mutation that leads to the most severe form of EBS. So it's an identical motif, and that was the reason this was chosen. And this is what one sees to the normal filaments in mice, control mice that overexpress the wild-type keratin 18, as compared with these mice. It's arginine 90 without the first methionine, arginine 90 with the first methionine, I'm sorry, 89 without the first methionine, but you can see complete disruption of the keratin cytoskeleton. As an approach to try to understand the function of these proteins, knowing that, of course, that PTMs at that time, phosphorylation was known of a number of intermediate filament proteins. We did quite a bit of work identifying a number of other post-translational modification, including glycosylation. Initially, we showed it for keratins 8 and 18, then it was shown for other intermediate filament proteins. There's also assimilation. But basically, phosphorylation is a major keratin, in all intermediate filament proteins are major targets of a number of different kinases. And I mentioned the phosphate sponge. They're very abundant. Under basal conditions, the overall stoichiometrically of phosphorylation is very low. But then any kind of a stress leads to hyperphosphorylation of any intermediate filament protein, and that affects the solubility of the intermediate filament protein. It affects the organization, which is critical, because when you have disruption of any keratin or other intermediate filament protein cytoskeleton, these cells are much more susceptible to necrosis, apoptosis, and also phosphorylation regulates the interaction with a number of different proteins that I won't have time to get into. So this mouse work led us then to to test whether keratin variants, mutations, 8 and 18 specifically, are associated with human disease. And we identified a number of different variants. And the bottom line here is that keratins 8 and 18, and actually keratin 19, we also showed variants, mutations in these keratins predisposed to disease. So it's the major hit, but one needs another hit, whether it's a viral infection, overexposure to alcohol, other metabolic genetic disorders that can then lead to end-stage liver disease. And this highlights the importance of phosphorylation just of keratin 8. We had identified this phosphorylation site. There's a fairly common variant about in 1% of the population or so, in those who actually have liver disease versus the normal distribution in about maybe 0.2, 0.3%. This particular variant here, glycine to cysteine, actually inhibits this phosphorylation site, which is essential for allowing the keratins to cope with stress. If this site cannot be phosphorylated, the hepatocyte doesn't like it. So knowing, of course, that the simple epithelial keratins mutations don't cause disease, we knew a lot about them. We had expression cell systems, mice, and things of that sort. So we thought we would actually target those cells using high throughput drug screening to see if we can identify small molecules that can normalize initially the disruption of simple epithelial keratins, and then test whether those particular small molecules can also have an effect in epidermal keratins and potentially other intermediate filament proteins. And this work was started by Raymond Kwan and Liu Chen in the lab, and then others who joined the lab continued the work. But the simple hypothesis is a high throughput drug screening, if one has the right assay, can hopefully, we know that when normal filaments are disrupted like this, whether it's in the cytoplasm or whether it's in the nucleus, the cell doesn't like it for a number of reasons, whether it's a cytoplasmic compartment or the nuclear compartment, and there's also a crosstalk between these two major compartments. So if we can identify compounds that somehow maybe modify the regulation of these disrupted keratins, and maybe can take the distribution of the filaments from here to here, maybe that could have a beneficial effect. So we use this well-known characterized libraries. We started with a kinase library, knowing that these proteins are phosphorylated, and you can use a 96 well plate. We use 384 well plates. We use control cells that, so these cells express GFP keratin 18. And so basically all the cells that express the keratin are going to be green. Normal cells are going to have a filamentous distribution, whereas the cells that express the mutant keratin 18, as predicted, are going to have this total disruption. So the positive hits are going to be the dark green, and this is basically an imaging system that's taught to recognize cells that have more of the more of green rather than the loss of green, which is either dead cells or cells that have the normal that basically have not been affected by the drug. And that's how we picked up a couple of hits. The first hit that we focused on was a small molecule called protein kinase PKC 412. And it turns out that actually this drug is used as a chemotherapeutic, as a pill. And I will talk about that in just one slide. So, but basically, like any other screen, sometimes we multiplex also these wells. In this initial case, we did not. So one needs to validate, and this is just a simple validation experiment. And you can see that this is a dose response, and that you can hopefully appreciate the difference of keratin distribution between the cells in this row versus the cells in the top row. So what's PKC412? For those who may not be familiar with this small molecule, it's a pan-kinase inhibitor. It has more selectivity towards PKC, but it also inhibits other kinases. For example, VEG receptor 2. It also inhibits a receptor called FLT3, tyrosine kinase. And this is important because it's mutated in about 1/3 of cases of AML. And that's one of the reasons why now it's in the United States, it's FDA approved as a treatment for acute myelocytic leukemia in patients who have FLT mutations where this kinase is activated. It's also used in systemic mastocytosis, which is caused by mutations in KIT. So we have the mice that were generated by NAMON. So we know that PKC412 works in cell systems, culture cell systems, but does it actually have any effect in an organ? And in this case, the liver. And the reason the liver, in fact, is the organ in patients who have variants in keratins 8 or 18 is, I mentioned at the top of the presentation, that hepatocytes, adult hepatocytes are unique, that they only have keratins 8 and 18. Other simple type epithelial cells, they have other keratins, and we showed using genetic models that these other keratins can functionally also compensate, as is the case in the intestine, by developing different kind of transgenic mice. So the evidence is pretty strong that the reason there's this massive disruption and impact on hepatocytes in the liver is because of this 1-to-1 ratio. And in the present, when mice are administered PKC412, the keratin is normalized in hepatocytes. The extent of liver injury, as determined by a serum test, transaminase, also normalized. And hopefully, you can appreciate here the phenotype in the mice is hemorrhage. Can be macroscopic, but usually it's microscopic, as you can see here. And you can see a significant improvement of the loss of hemorrhage in these animals, in addition to other histologic features. In another screen, the reason I'm showing you these is because these are actually drugs then that we're going to be, that I will show you some data in terms of their potential use in EBS. Another compound that we picked up is a compound called PP2. It's actually used in a lot of studies, but it's not used in any patients because of its potential toxicity. It's a SRC tyrosine kinase inhibitor. And then another compound I will be talking to you about is called vemurafenib, and that's a serine threonine BRAF kinase inhibitor, and it will hopefully make sense why I'm mentioning those. So we identified PP2. Again, same type of, it was a different library, but basically the same approach. Do the validation in cells, then move to our experimental animal system. And in this case, because of the metabolism of the drug, which Lee shows in her publication, its metabolism plays a role in that the phenotype is primarily in males, as you can see here, but not in females. So, for example, one induces liver injury in the animals. You can see there's a massive hemorrhage, but in the presence of PP2, after inducing hemorrhage by administering fast ligand to these animals, you can see there's a dramatic decrease in the hemorrhage, improvement in histology, and also serologic improvement as well. So the mechanism then, another important thing after identifying a drug, and that's actually is one of the benefits of the screening anyway, is that we were able to pick up, because we studied in depth how does PKC work. It works by dephosphorylation, but it's not really so much dephosphorylation of the keratins that it affects. It actually leads to dephosphorylation of non-muscle myosin heavy chain, and then that dephosphorylation leads to an association with the keratins, and this is shown here by immunoprecipitation. So you can see that in cells treated with PKC412, there's more of an association with the keratins, and this also correlates with normalization of the keratin filaments. By contrast, this PP2 compound leads to dephosphorylation, as shown here by mass spec, about 50% of the phosphorylation, overall phosphorylation of keratin 18, 60% of keratin 8, and we have generated different phosphospecific antibodies, and you can see there is significant dephosphorylation of keratin 18 at two different sites, and also of keratin 8. So the way these compounds are working, I gave you some information about PKC412. It leads to dephosphorylation of this myosin, non-muscle myosin. In the case of PP2, it leads to dephosphorylation of the keratins. Now, it's known already that PP2 inhibits SRC. And it's also known that SRC actually inhibits RAF kinase. And we had shown, in fact, that RAF is a physiologic kinase for the phosphorylation of keratins 8 and 18. And it's already known that there's an inhibitor called vemurafenib that's used in the treatment of patients with melanoma as a RAF kinase inhibitor. So then the obvious question is, does vemurafenib phenocopy the effect of PP2? Because then vemurafenib could potentially be used as a therapeutic in patients with EBS. And the answer is yes. So again, this is in this case using our animal experimental system, because we want to look physiologically in the context of a whole tissue rather than individual cells. And the same thing here, in the presence of absence of vemurafenib, this is the extent of hemorrhage, maximal hemorrhage. In the presence of low dose of vemurafenib, there is a massive improvement. Higher doses, so we did a dose response, it becomes toxic and it no longer has an effect. And you can see the effect here. It's in again, it's in male because of the metabolism of the drug. You can see that there is a significant decrease in apoptosis as determined by immunoblotting with looking for cleaved caspase 3. This is only in male, but not so much in female. If you give the high dose, actually, it causes even more cell death by apoptosis. And the same thing for caspase 7, and this is just a Comanche stain for control. So this is now where the liver actually took us to the skin. And here we had already collaborated with Thomas Megan's lab, looking while we're doing studies related to the function of simple epithelial keratins using some of the genetic models that Thomas had generated. So Katrin, at that time, a postdoc in Thomas's lab, actually she came to the University of Michigan. She spent 3 months working in our lab doing high throughput drug screening. And she has 2 other potentially very exciting compounds that still need investigation. But she was interested in looking more closely at PKC412 because of the results that I shared with you. And sure enough, PKC412 in cell lines that are derived from patients with EBS, this is the cell line that expresses the most severe mutation that leads to severe form of EBS. And this is now treating those cells with PKC412. You can see that the extent of aggregation is really tremendous as measured by immunofluorescence staining. Another, of course, assay that's very commonly used that I think maybe many of you on the call are more familiar with it than I am, but we certainly have used it, is a sheet fragmentation assay. Again, taking the cell system, just simply shaking the cells to normal human keratinocytes after rotation. You can get some breakage, if you will, of that sheet into fragments. However, in the presence of the mutation, this EBS patient mutation, you can see significant breakage, disruption of this sheet. However, in the presence of PKC412, you can see significant resistance to disruption of the epithelial sheet in the presence of PKC412. Katrina went on she was mentoring a graduate student, who is now Dr. Job, and looked at other keratin mutations, another keratin 14 mutations, keratin 5 mutations, and as you can also appreciate the effect of protection. This is in the presence of PKC12 as compared with a carrier, which is DMSO. There's also protection that was shown in the same manuscript that was published last year, and this is mimics, if you will, epidermal. It's called epidermal equivalence, where you have multi-layer formation of cells, whether they're normal human keratinocytes or they're mutant keratin 14 or keratin 5. These cells are seeded and then followed, and they're harvested after 8 days, and you can actually do histologic analysis, biochemical analysis, and hopefully you can appreciate in the case of that there's what would be the equivalent of a blister formation in the sheets from the K14 mutant and also from the K5 mutant, but those sheets disappear, those blisters, if you will, blister equivalents disappear in the presence of PKC412 in the two keratin mutations. So our findings in vemurafenib and the PKC412, which work by totally different mechanisms, raise the hypothesis that could one use them as a mix, as a combination, as a cocktail, if you will. The potential advantage of this is that if they truly work in vivo by different mechanisms, then using lower concentrations by their use as a mixture might decrease the toxicity, and potentially they may act synergistically. And especially in this case, we have drugs that could be repurposed. So that was kind of the hypothesis that we had proposed in the review, but also it's based on Pei Li's work where she identified PP2, showed the association of that took us to vemurafenib as a potential drug that could be repurposed. So we started again using our cell system. And this is cells that express mutant keratin 18. I'm going to be then moving to keratin 14, but just to see does this system have any effect in the cell system and also in the animals where these inhibitors were identified and shown to work in the first place. And you can see here that this is the dose that we had used as individual compounds. And this is half dose of PKC412 and vemurafenib, and this is using basically doubling the dose. So the effect is basically using half the dose is just as effective, if you will, at least in terms of protection from apoptosis, as compared with using a single individual doses. If you take it now to the whole animal, and these are the mice that were developed that we've actually shared with a number of different laboratories that express the mutant keratin 18 in simple type epithelia, with the liver being the primary target, just like in humans. You can see here that one can induce liver damage by administering Fas ligand, and you get by tunnel staining you can appreciate significant apoptosis that hopefully you can appreciate and is quantified here. When you use PKC412, it's much less. PP2, vemurafenib, much less as predicted. But when you combine them, this is really the difference here. Combining the drugs at the lower dose, you can also see the same beneficial effect, at least for keratin 18. So, of course, these are kinase inhibitors. So how are they working also in cells or in the liver? And interesting finding here is that depending on the kinase inhibitor, whether you're looking at, for example, at this is here the MAP kinase is looking at phosphorylation of ERK, activation of ERK, Jun kinase or P38, the effects are either overlapping or unique. So the prediction here is that when you use a combination, you're more likely to kind of cover the inhibition of more kinases and potentially have more of a beneficial effect. But of course, the concern would be a potential toxic effect. So this preclinical work, then we wanted to see whether we could take it to patients. And the goal is to, if we're going to take it to patients, of course, it's a skin disease. They can take the drug orally, more predictably, more side effects. PKC412 is used as a oral drug to treat acute myelocytic leukemia. But if it could be used as a skin, the prediction is that the toxicity would be much less if we have it as a cream. And here we collaborated with Dr. Mishnia Cohn, who's a well-known skin biologist whose expertise actually is testing, developing ointments, creams, and so on. And we jointly, Bo and myself, mentored one of our graduate students who are interested in this project, who was working with Pei Li, who has done a lot of work characterizing these specific compounds. So the cream was developed, and at least a cream to test. And we have 3 creams. So one cream has PKC412, the other cream has vemurafenib, and the third cream has a cocktail. We tested in cadaver. I'm not showing you the data here. The compounds concentrate more in the epidermis as compared to the dermis, and there's really no detectable flow through based on the cadaver skin system, experimental system that's used. But unfortunately, as one can imagine, in cadavers, it depends on how long the skin has been isolated. Many times it's 12 to up to 24 or more hours. It's not a good system to look at phosphorylation, to look at biologic activity. So fortunately, there's a commercial source in France that provides surgical specimens of a trademark called GenoSkin that any one of us can use to have more of a physiologic human in vivo effect for particular compounds. In this case, we're looking at a cream. So so far we've done 3 patients. We're actually doing actively, as I'm talking, an experiment on 6 additional patients. But basically, the simple experiment here is to use control cream, PKC412 cream, vemurafenib cream, or the cocktail cream that I mentioned, the 3 creams. The retention of the drug was measured by mass spectrometry. It seems small, but apparently talking to Bo, who's an expert in cream and ointment formulations, this is reasonable. So at least there's some in the epidermis, there's some in the dermis. We're repeating these numbers. The surprise result is that actually when you have the cocktail, the PKC412 lower dose, for whatever reason, at least in a triplicate, the retention in the epidermis is higher than when we use twice the dose of PKC412. And the same thing is for the vemurafenib. And these data need to be replicated, and we have some reasons why this might be the case. So now, what is the effect on GenoSkin of these compounds, either singly, PKC412 vemurafenib, or the cocktail? And this is a biochemical assessment here, and this is a proteomic assessment. In terms of looking at phosphorylation, this is a phosphoserine threonine antibody that a lot of investigators use, and hopefully you can appreciate that a number of proteins become hypophosphorylated. And we knew this before from the liver case, for example. In this case, it's human skin, and also the same is noted with the cocktail. More likely, at least depending on the protein, because of PKC412 rather than vemurafenib. The interesting with vemurafenib is that it actually led to an increase in some of the collagens, which is known to be protective in the skin. And that was statistically significant and reproducible. Depending on the collagen, it was one of the collagens was with PKC412, the other collagen was with a cocktail, with the trend being vemurafenib. Using a proteomic approach, PKC412 led to the overall dephosphorylation as predicted, but this is verified here, and also led to a decrease in phosphorylation of K14. This is K5, I'm sorry, this is K14 with vemurafenib. And there is some also dephosphorylation of keratin 1. At least by proteomic approach, we did not pick up any dephosphorylation of keratin 10. So where are we at? The next step really is continuing a collaboration with Professor Haas, who's at the University of Freiburg. She oversees a referral center in Germany, but also neighboring countries of patients with EBS. So the initial experiment, we're just completing the paperwork. We have the cream that we would like to at least try on a compassionate basis, is to, just because of the limited number of patients, it's a compassionate basis, is to try the PKC412, try the cocktail, and compare this to a vehicle. So one arm, one leg would be a vehicle, which is shown in the green, and the other arm on leg would either be PKC412 or a cocktail. So that's the planned experiment for hopefully we'll be able to do this during the first half of 2026. Need to special thanks to Sagir for inviting me to give this presentation. Of course, thanks to DEBRA UK for everything that they do for patients. Thanks to all the donors, of course, that make the research grants possible that DEBRA is able to give. Without this, of course, a lot of the advancements that are being made in EB. In this case, I'm talking, hopefully there'll be some funding of EBS that makes this possible. And I wouldn't be giving you this presentation. A lot of the work that was keratin was done by many members of the lab that I was blessed to work with as teammates, as co-workers. And this is our current lab here at Rutgers University. Lots of collaborators. I'm just showing in bold here the folks, collaborators, and also co-workers in the lab. For example, Lu Chen, Raymond Kwan now is his own position, but he's still working on a couple of manuscripts. And also we're continuing to work with Katrin. I mentioned Hannah, graduate student who has developed the cream, and also is working together with Pei to test the cream. Thank you so much. Maybe I can stop sharing. What do you recommend, Sagir, here? Should I stop sharing?
Yeah, it means just stop sharing.
Okay.
Yeah, and I guess the floor is now open for a Q&A session. So please submit your questions in the Q&A function or in the webinar chat room, and then I can raise them with Bishr when we'll try and answer them as best as we can. So while people are busy typing away, I've got a question. What are you, from a clinical point of view, what are you expecting from the creams that you're looking to test beginning of next year?
So the participants would basically one is just purely symptoms, pain, itching, skin sensitivity. Of course, the cream itself can cause skin sensitivity, but that's one thing that we would want to exclude with a control cream. But basically, the other is a decrease in blister formation when we follow those patients with time. And that's really going to be particularly, weight-bearing areas, things of that sort, where can they now do more weight-bearing, exercise more, things of that sort. So that's going to be an important readout. There will be no skin biopsies in the beginning, but initially just a clinical assessment, and then if it's positive, then we would have more of a justification to consider anyway, seeking permission for either a brushing of the skin or a skin biopsy.
Yeah, we've not had any questions coming from you, so I'll continue asking the questions. Can you see the Q&A session?
I can see. Let's see. Yeah, I can. Let me see if I can. Yeah.
Yeah. So, no questions at the moment. With these MAP kinase inhibitors, what do you think is the downstream targets that potentially could be helping with the reduced potentially blisters and wounds that you might see with a cream?
Yeah, so here, at least based on the work that we've done, there are two types of targets, but there's likely to be more. But two
Yeah,
major targets stoichiometrically are the keratins themselves,
yeah,
K14 and K5,
mm,
but also keratin binding proteins, for example,
mm,
non-muscle myosin heavy chains.
yeah,
Desmoplakin is another one. So there's and we can
mm,
also identify at least dephosphorylated proteins that are known to bind to keratins or
okay,
to help stabilize, the plasma membrane, or some nuclear proteins as well. So I mean, the signaling pathways are really multiple in this case,
yeah.
but presumably, predictably, there's a couple of major ones, and that's why
Mm.
we're seeing the phenotype.
We've got a couple of questions that come through. The changes in phosphorylations of the keratin was fairly small, even with the combination treatment. What do you think are the main targets in the skin?
Well, so in this case, I mean, I think the key experiment is really going to be in the patients. But in the cell systems, 50% decrease in phosphorylation is actually significant. And many times, depending on the site and the location, all you really need, because that's affecting, for example, a binding protein that regulates a number of kinases like 14-3-3, for example. So stoichiometrically, it doesn't have to decrease by 50% actually is a robust, it would be considered a robust effect in the realm of phosphorylation. But of course, it's site specific, and it depends on what the site that's being phosphorylated.
Yeah. And then we've got another question that's coming. Congratulations, great work. Can you explain the female and male differences, and also if it's known in patients?
Yeah. So the male-female difference that's actually encountered this sexual dimorphism in a number of studies, rodent studies, and the reason for that, and we validated this, is the cytochrome P450 expression profiles in male as compared with female animals. And we actually show we can measure metabolites of those drugs and show that the differences in metabolism in male versus female. And we know, for example, whether it's the prodrug is the active drug or the metabolite is the active drug. So that's really the reason. I mean, it's an important and that's one important reason to test both male and female animals,
Yeah.
as now the NIH actually expects and requires. I'm not sure how it is like in the UK, but in other countries and also in, say, in
Yeah.
Europe.
Yeah. Just kind of following on to that question, do women have more severe EB simplex than men?
I asked Dr. Haas this question, and maybe others dermatologists on this call can correct me, but in general, no. But
Hmm.
but I'm not sure. For example, if they stress, like for example, if they do more blisters like in their feet or things of
Hmm.
that sort with stress, male versus female. That was a question I posed to Dr. Haas, and the answer was
Yeah.
no.
I don't think we've got any more
I hope
questions
I'm, I
coming.
hope I'm quoting her correctly. Otherwise, she's going to be angry with me.
We'll edit this part of the webinar out for
All
you.
right. All right.
And, no, I don't think we've got any more questions coming through. So on that note, there's no further questions. We'll stop the Q&A function. Uh, so before everybody leaves, I'll quickly share my screen.
And if anyone, they can always, I'm happy that people can reach out to me with any questions. I'm more happy to, any questions come up later. I'm more than happy. We're happy to share reagents and things
Yeah,
of that sort. So,
hopefully you can see my screen.
yeah.
Yeah, perfect. So just like to say, our next webinar, it will be in January 2026. That still shocks me that we're coming to the end of 2025 and obviously no rest for the wicked. So we've organized the next webinar for Wednesday, the 14th of January. We're lucky to have Dr. Christine Saltman based in Zurich, and she will be talking about strengthening and signaling proteins in skin cells. Before I leave you, I'd just like to wish everybody a Merry Christmas. I know it's early, but it's probably my only opportunity to tell you this. So again, thank you very much to join the webinar. Have a lovely break, and we will see you in the new year. And on that note, thank you very much and have a good day.